So thank you for staying here despite your starving stomach. We've heard during this conference a lot that how standardization was important for the MPS, and also thanks to the great introduction that I had from the previous speakers. And today I would like to show you now an example of an automated platform that enables to automate the perfusion of engineered tissue. And this work was performed at CSCM and also with our collaboration partner from Utrecht University. So we also heard many times at the conference that vascularization is one of the major challenges today for building good models and tissue engineering. So no matter whether we have 3D cell models, tissue engineering, microphysiological systems, we need, in all cases, to bring oxygen and nutrients to the tissue. At the same time, as mentioned before, we need to standardize our technologies. We need to have more reliable data. We need to parallelize the data to increase the throughput. And for that, many researchers are using the multi-well plate format, as we also saw just before, as they are quite convenient to parallelize the experiments. And in order to well parallelize and increase the throughput, automation is also needed to bring the technology, these models to the step further. At CSCM, we address this with our Smart Leads program. So I also had it was nice to have already an introduction from Sandro in his talk about the Smart Leads. We are developing the Smart Lead in order to automate, to bring automation onto the multi-well plate format. The principle is to rely on a commercial base plate, could be a normal base plate or also a specialized base plate with a multi-well plate format. And we bring fluidic function onto the lid with microfluidic channels in the lid to bring liquid into the wells and from the well, and therefore enabling automation of cell culture processes. The example that you can see here on this picture is a smart lid that was developed for 24 well plate format and that enables automated medium exchange. So thanks to the inlet and outlet port, we can remove half of the medium in the cell culture and then bring in all wells at the same time a new medium, thereby automating the medium exchange without having to remove the lid during the cell culture. And we can also bring other functions to this smart lid, such as perfusion, pooling, or sampling and sensing, as mentioned by Sandro before. And today, I would like to present you the perfusion function that we can address by using this smart lid technology. If we want to reproduce the physiological microenvironment, we need to perfuse, we need to flow liquid in the tissue. And as in the body, we have unidirectional perfusion. So we want to focus on unidirectional perfusion in a well plate here. And also, ideally, in the case of tissue, we would like to also perfuse through a tissue. So to have a tissue in a closed chamber and addressing the perfusion. Now, to show you that, I will use one of our European projects, OrganTrans, where you can see the different partners working the project here below, if you can see it. And the goal of this project is to fabricate liver construct for liver regenerative therapies. It is based on using organoids and combining them with bioprinting approaches to create the liver tissue. If you want more information about the project, I invite you also to visit the website and or to come to me after the talk if you want more information about the project. As today we'll really concentrate on how we can move from a printed bioconstruct into a functional tissue. And for that, we need to mature the tissue. We need to perfuse the tissue. when we bioprint the tissue, we need also, we don't see it well, we need a specific we insert to hold the tissue, to print the tissue inside, and then to transport the tissue into the perfusion platform. For the perfusion, we use the smart lid technology that I showed you before, and then enable the perfusion from days to weeks. And finally, we get to a functional tissue that we can test in vitro and also transplant in vivo. But now we'll really concentrate on this maturation process. And here you see the system, how the system for the perfusion looks like, how we can create this closed chamber to perfuse unidirectionally into the wells. So first we have the labware, so we have the standard 6-well plate. And then we have the smart lid here with the microfluidic channels, where here in this case we have 6 inlet channels to bring the liquid in the wells and 1 outlet channel to remove the liquid and then lead to this flow. We have a custom insert. You can see the insert in the well plate to hold the bioprinted construct, which is in this case 1 by 1 centimeter by half a centimeter high. To create the closed chamber, we have a gasket that comes between the insert and the lid. And all the materials are biocompatible and sterilizable. And then we want to control the flow. So we want to control the flow unidirectionally, and we want to be able also to change the flow rate, as this can change depending on the tissue. We use a multi-channel peristaltic pump that enables parallelization of the flow in the different wells, and where the media are not in contact with the pump. And for the control of the environment, we can use a standard incubator. So these are photographs of the labware that we have developed. So here you have the smart lid. It's an injection molded smart lid in one piece with the 6 inlet and 1 outlet and microchannels on the top of the lid. And here you have the insert that was specifically developed to hold the construct. And these labware are integrated into the platform, which is an automated perfusion platform. So you can maybe see that the lid is placed here with the insert below. In this case, you can see also the gel, which is placed in the insert. And then we have the pump, the peristaltic pump here that is integrated on the platform. We have the media solution, so we can run the platform either in flow-through mode or in recirculation mode, depending on the need. And we have dedicated electronics that enable to tune the flow that you want in the different chambers. And the whole platform can be transported onto a microscope stage to image the wells, or it can be placed also in the incubator to run the perfusion to control the environment. And this is what you can see here, the perfusion platform in the incubator for the experiment, and the 6 gels in the well plate that can be perfused in parallel. And in this case, we have collagen construct with cells, and we measured the cell viability after different times of perfusion, showing that after perfusion we have a better viability of the cells. Of course, this depends also on the type of construct we want. And now I want to go back to the example that I introduced before with the organ trans technology, where in this case we use bioprinting to make our tissue. So for the bioprinting, we collaborated with Regenue, which are a provider of a bioprinting platform in Switzerland, and we developed this dynamic bioprinting device that can print a mixture of gels and spheroids to make the gel. And this leads to the bioprinting construct that you can see here, which has the size of 1 centimeter and half a centimeter high. And the latest structure shows spheroids embedded in the gel. And we have also sacrificial channels, which were printed and that can be removed afterwards, and that facilitate the perfusion of the liquid through the construct. So now we are interested in how this gel can vascularize, and so here you can see how the setup placed in the incubator at our partner in Utrecht. And then we have on the one side the formation of macro vessels, which are provided by the printing technology. So by dissolving these sacrificial channels, we can see on the bottom image here endothelial cells labeled with GFP that can circulate in these hollow channels. And the little dots here are actually the spheroids that were printed with the gel. And at the same time, we have the formation of microvessels, this microvascularization formed by, thanks to the composition of the spheroids, which are tricultural spheroids, comprising also endothelial cells, stem cells, and hepatic cells. And here you can see sprouting from the endothelial cells from the organoids. And on the bottom image, you can see the formation of the network from the endothelial cells present in the gel. So we have really this combination of both vascularization. And then, if you look here at the viability and how the tissue looks like after 1 week of in vitro maturation in the platform, you can see before maturation here we still have the sacrificial channels, we have the organoids, and after 1 week on the right-hand side here, you see the spheroids starts really, you see the sprouting of the spheroids with the cells going out of the spheroids, and that's the viability of the cells with the live dead cells. You see that the cells are well alive after 1 week in vitro maturation. And there are also in vivo transplantation tests being performed by our partner at Utrecht University, when you can see after 1 week implantation of the liver construct in the mouse, that the spheroids are still quite visible, but they really start to create this vasculature, and after 4 weeks, that the construct is well populated with cells. So this shows the automated platform that enables the perfusion of bioengineered tissue. So it's a platform which is quite versatile, which fits to the standard multi-well plate format, and we can customize the different parts according to the needs. In this case, we customized the insert for the bioprinting approach that we had. So we can have a tunable flow and unidirectional. So it enables automation and parallelization of the perfusion, and the control environments are provided by the incubator. So we could show vascularization and tissue viability, and how this system can provide throughput standardization for the scalability of the maturation of tissue. So this is an example of a technology that we are developing at CSEM. CSEM is actually a research center, research company in Switzerland with the mission to transfer technology to industry, so to adapt also to use our technology to fit and address your needs. And in particular for the life science, we are aiming at standardizing the life science processes by developing tools for the life science based on different technological pillars, so to the cell microsystems to really create this microphysiological environment for the cells. We also have some biomonitoring activities, and we use lab automation and artificial intelligence to really bring the technologies to the next level. So if you are interested in such technology or to know more about what I've presented, we have a booth here, which is the booth number 40 at the end of the room on the left. So please don't hesitate to come and visit us. We can also show you some other technologies that we have developed or discuss some ideas for collaboration. We collaborate with academics and with industry. We had posters presented yesterday, but still, if you still have a chance to see them to see the technologies at the booth. And finally, I want to invite you to this workshop that we're organizing, the Next Gen Organ and Chip conference that will take place in Switzerland, in Zurich, 24th and 25th of August, where it's a great occasion, great meeting between the research and industry to bring the nice results from research into industrializable technologies. And with that, I thank you for your attention.
Thank you, Stephanie. Do we have any questions?
Thank you for your talk. I'm just wondering how you create actually the liver organoid. What types of cells used to create the liver organoid?
What the cells are used to create the spheroids, you mean? The liver organoid. There are 3 types of cells. There are the hepatic cells, chondrocyte, and there are the mesenchymal stem cells and the endothelial cells, so UVEX. So it's, the way we did was we used standardized plate from the company Kugelmeyers, where you have microwells and you mix the cells in the defined ratios, and then you can form the organoids in a very standardized way. Yes, I mean, I didn't present that. I focused really the talk on the automation for the perfusion, and all the functionality of the organoids was assessed by our partner in Utrecht University. They have assessed the functionality of the spheroids individually and also of the spheroids inside the tissue, where they looked at hepatic functional markers. So if you want to know more, I can give you the contact also from our partners in Utrecht. It's the group of Boder-Pasche, and they're very specialized into this topic.
Next question here.
Hi, thanks for your talk. Lisa Mojanics from Elvis's Microfluidics Innovation Center. So I'm interested if you can comment a little bit more about the recirculation mode of your lid, because it looks like you had 6 inlets and only 1 common outlet, and in particular, if the liquid mixes during the recirculation between the different wells. Thanks.
Yeah. Okay, so for the recirculation in this configuration, the goal was to parallelize the same tissues. We didn't care about mixing of the medium, so we have 6 inlets, but as we have only 1 outlet, when we do the recirculation, all the outlet is coming back into the same bottle. So in this case, it is mixing the medium. Now, with the technology, we could also further customize the lid to have 6 inlet and 6 outlet, which would be quite easily feasible if we don't want mixing between the different wells.
Sorry, thank you for the talk. Maybe I missed this point, but when you grow the spheroids by using the 3D bioprinting, which kind of polymers, which kind of material did you use to grow the spheroids?
So to grow the spheroid, I didn't show this part yet. So I used we used the multi-well plate from a multi-well plate. It's like the Euler plate, but it's another approach from Kugelmeyers, which are inverted pyramid coated, it's coated plastics. And then we embedded the spheroids in a PEG gel. Was it your question for the embedding of the spheroid in the tissue or for the formation?
The biomaterials, yeah. The PEG you use,
the
PG.
So yeah, it's a PEG-based polymer that is developed at DWI by the group of Laura Delaporte, and they use it's a half degradable PEG, so you have some degradable entities also that will degrade after implantation of the tissue in the body. So we use a combination of MMP PEGs and I think, vinyl sulfone PEG. I don't remember exactly. Sorry.
Okay, thank you.
So I think it's time to end the session. Thank you very much for speakers. Thank you for the audience for the questions, and bon appétit. Have a nice lunch.